अल्पना जी, वेबसाइट में लिंक अपडेट हो गया।
ओके ओके
Yeah, let us start the program.
Okay, sure. So, welcome everyone to our next seminar for Science Setu. This event is being organized as a part of Azadi Ka Amrit Utsav, which is an initiative by the Government of India to commemorate 75 years of Indian independence, its glorious history and our achievements. Now, this Amrit Utsav was started in the month of March this year and will continue for the next 75 weeks till August 2023. Now, with this short introduction, I'd like to request Dr. Priyadarshi Basu to please introduce our speaker for today.
Thank you, Alpana. Can you hear me?
Yes, yes.
Good morning, everyone, and welcome to the second Science Setu webinar from NIPMG. And today's speaker is Dr. Moulinath Acharya. He did his master's from Banaras Hindu University with zoology, and then his PhD from CSIR Indian Institute of Chemical Biology. His thesis title was Molecular Characterization of Primary Open-Angle Glaucoma in Indian patients. Following that, Dr. Acharya went for a postdoc and he did his postdoc from Department of Biological Sciences and Center for Prions and Protein Folding Diseases at University of Alberta in Canada. He did a second postdoc in the same university in the Department of Medical Genetics. In 2012, he joined as a faculty member of National Institute of Biomedical Genomics. Dr. Acharya was selected as member of the Indian National Young Academy of Sciences, which is an initiative of the Indian National Science Academy for a period of 5 years. His lab is interested in, is focused primarily on genomics of complex traits affecting vision and neurodegeneration, and also investigating genetic heterogeneity in rare diseases with neuronal defects through integration of relevant concepts and knowledge that include high throughput omics analysis amalgamated with appropriate in silico and functional genomics approach. So I would also like to add that after when the lecture starts, you may type your questions in the chat box of YouTube. Type the questions there and they'll be addressed at the end of the seminar. So thank you, and over to you, Dr. Acharya.
Yeah, hello, welcome to NIPMG webinar for the Science Setu. I'm thankful to Dr. Priyadarshi Basu for the kind introduction, and I hope that all of you and your family members and near and dear ones are safe and healthy during this testing time of this pandemic. So without further delay, I would like to start my presentation today. Let me share the slides. So, can you see my slide?
Yes.
Yes, we can
Okay.
see.
Yeah, so today's topic, as Dr. Priyadarshi Basu mentioned, genomics of rare congenital disorders, challenges and achievements. The first slide is basically, I would like to give some introduction about rare diseases. You can see this, this is borrowed from the Rare Disease India initiative, where India has a burden of 70 million rare disease patients, as you can see it here. And then if we move, there are children and rare diseases. Approximately 50% of the people affected by rare diseases are children, and 30% of the children with rare diseases will not live to see their 5th birthday. So this is an alarming fact. So here are a few quick facts about rare diseases. There are 7,000 known rare diseases. 1 in 20 Indian patients diagnosed with rare disease. 350 million rare disease patients globally, which is a big number. Average of 7 years to diagnose a rare disease. 80% of rare disease have identifiable genetic origin, which we will be talking about today. And rare disease include inherited cancers, autoimmune disorders, congenital malformations, and sometimes rare infectious diseases. Today's talk will be focused on the congenital genetic diseases only. If we look at rare disease definition, the total number of people having the disease, its prevalence, non-availability of treatment for the disorder. The World Health Organization has suggested that a rare disease should be defined as one with frequency less than 6.5 to 10 per 10,000 people. India, like many developing countries, currently has no standard definition. Considering the large population of India, Organization for Rare Disease suggests the disease to be defined as rare if it affects 1 in 5,000 people or less. The Orphan Drug Act in 1983 enacted first in US. This law is designed to facilitate the development and commercialization of drugs to treat rare diseases. The global orphan drugs market exceeded $50 billion in 2011. Among them, only around 500 approved orphan drugs exist in the market and over 100 in clinical trials. So majority of the diseases have no treatment when they exist are mostly unaffordable. I'll come to specific example in my next few slides. But before that, what should we do with the rare diseases in India? This whole part I have taken from a paper published in Current Science by former ICMR Director General Professor V.M. Katoj, Professor Partha Majumdar, and Professor Alok Bhattacharya. So what they said, this is basically the title of the paper, Rare Diseases Need Our Attention. The first thing is mapping and identification of patients with rare diseases and creation of a rare disease registry in the country. This will be helpful in formulating a policy on rare diseases. One of the major problems in rare diseases is the lack of awareness, not only among common people, but also among clinicians and researchers. A concerted effort should be made to increase awareness. We need to set up new centers and strengthen the already existing ones on molecular diagnosis of rare diseases, taking advantage of new low-cost technologies that are becoming available. A number of centers of excellence should be set up as referral and research centers for patient management and for developing therapeutics and diagnostics. In order to enhance the mobility of patients with rare diseases, we need to provide adequate assistive devices. This can be achieved through better designing using smart, lightweight materials. There should be a concerted effort to develop therapies for these diseases and make this accessible and available to all patients. For this, we need to enhance our capabilities in technology platforms such as animal model, gene therapy, stem cells, in addition to enhancing our abilities to identify therapies based on small molecules. A rare orphan disease policy is required to implement most of the goals outlined here. Currently, there is none in India through 23 countries such as US, UK, Taiwan, Ukraine, and many European countries have already formulated specific policies. However, there has been an attempt to formulate an orphan rare disease policy for the state of Karnataka. In addition to an enabling policy, the government needs to allocate resources specifically earmarked for rare diseases. And patient advocacy groups should be part of development and implementation of the policy, and their suggestions should be sought at every stage. With that, the government has already taken some initiatives. As you can see it here, the Ministry of Health and Family Welfare has formulated the National Health Policy 2017 that underscores the need for management of rare and orphan diseases. First comes prevention, awareness generation, training of doctors, funding support, that's most important, for treatment on the parameters to be defined by a central technical administrative committee, promotion of research and development for drugs for treatment of rare diseases and diagnostics at affordable prices, and measures for making the drugs for rare diseases more affordable. Finally, the strengthening of laboratory networks and development of centers of excellence. So you see that these are the policy initiatives. So if I go to my next slide, I will just show you one example of spinal muscular atrophy, which is basically a debilitating condition where you can see that the synaptic dysfunction loss of synapses, motor neuron hyperexcitability, and motor neuron death has been happening in the spinal cord and in the neuromuscular junction as well. So reduced myofiber size, synaptic dysfunction, impaired development, denervation. So the effect is very severe, and you can see a patient using a wheelchair in a wheelchair. So what is the cause of this disease? There is a gene called SMN1. This is survival of motor neuron. Is the telomeric copy of the gene encoding the SMN protein. The centromeric copy is termed SMN2. This SMN1 and SMN2 are part of a 500 kb inverted duplication on chromosome 5q13. This duplicated region contains at least 4 genes and repetitive elements which make it prone to rearrangements and deletions. So what happens? This SMN1 and SMN2 are nearly identical and encode the same protein. The critical sequence difference between the two is a single nucleotide in exon 7, which is thought to be an exon splice enhancer. So what happens in healthy individual? This SMN1 is fully functional, 100% full length SMN1. And you can see that SMN2 is also, even if the SMN2 is not functional, you will not have a problem. But for a SMN patient, the spinal muscular atrophy patient, this 0% for full length SMN1, because there is a mutation in exon 7, that makes it much more difficult. So 10% of the full length SMN2 or 90% of the deleted SMN2. So that actually the number of copies determine the severity of the disease, as you can see it here. As you can see, the increasement of these deletion 7, the disease severity increases. Now, what should we do with this? Okay, so there is a drug. This is called Sologen Sma or Sologen Sma. The genetic root cause of SMA is the missing of improper working of SMN1 gene that I have mentioned already. This is a gene therapy approved for children less than 2 years, is given as one-time infusion into the veins over 60 minutes, made up of new working copy of SMN gene and is given through a vector targeting the motor neuron cells in the body. The new gene delivered by the vector on reaching the destination tells the motor neuron cells to make SMN protein. So that's basically what it is. Again, this is not a cure and cannot reverse permanent damage already caused by spinal muscular atrophy before treatment. So it is very important to start the medication as early as possible. And that, how do you do that? By doing the genetic screening. By the way, why we need an orphan drug policy? Because single dose of these Sologen Sma costs 14 crores. So you can see that 99% of Indian, 95% to 99% of Indian people cannot afford this. Again, another important method was Antisense Oligotechnology that has been used. We've been using this for treatment of many rare diseases, mostly the neuromuscular diseases. So again, this is a short oligonucleotide capable of binding with RNA, eventually leading to knockout of the gene expression. You can see that usually we have the phosphodiester bonds in the DNA, which is replaced here by a morpholine ring. And this is basically, this is how this binds to the RNA, this phosphorodimidate intersubunit linkage replaces this anionic phosphodiester linkage, so it stalls there. So the mRNA stalls, it cannot make the protein. So this antisense therapy has been used for many movement disorders, starting from spinal muscular atrophy, Duchenne muscular dystrophy, Huntington, Alzheimer's, amyotrophic lateral sclerosis, Parkinson, and spinocerebellar ataxia. So where do we start from here? I mean, the rare disease actually needs our attention. Our means the geneticist's attention. So let us have a look at the genomic architecture of these rare genetic diseases. If we, not the rare genetic diseases as such. So if you look at the Mendelian diseases, we have rare, simple, monogenic, having mostly coding mutations. And then we have non-Mendelians also, as you can see, that common, complex, multigenic, conjugated with environmental effect. They are having mostly non-coding mutations. Now, if we talk about the mutations or rare diseases, we should also look at this fantastic database, which is called Online Mendelian Inheritance in Man, or OMIM, which is a comprehensive authoritative compendium of human genes and genetic phenotypes that is freely available and updated daily. This database was initiated in the early 1960s by Dr. Victor McKusick as a catalog of Mendelian traits and disorders entitled Mendelian Inheritance in Man. So 12 book editions of MIM were published between 1966 to 1998. The online version was created in 1985 by a collaboration between the NLM, the National Library of Medicine, and William Welch Medical Library at Johns Hopkins. It was made generally available on the internet starting 1987. 1995, OMIM was developed for the World Wide Web by NCBI, the National Center for Biotechnology Information. OMIM is authored and edited at the McKusick Nathan Institute of Genetic Medicine at Johns Hopkins University. And this is basically the paper. You can, if you are interested, you can go back and read this paper. So this is basically derived from OMIM database. If you look at the Mendelian diseases and traits, the suspected Mendelian diseases, this is a little bit older, is 1900, where molecular basis known is almost 3500 and molecular basis unknown is 1800. So if we look at this distribution of phenotypes across genes, we can see that maximum in OMIM, the maximum number of genes with one phenotype. That is one gene, one phenotype, okay? But there are diseases that has two phenotypes. There are genes that with two phenotypes, three phenotypes, or more than four phenotypes. And that is basically depicted here also. The same thing, single gene disorders and traits. Here the blue is phenotypes and the amber is gene. You can see that susceptibility to complex diseases of infection is also there. Some genes may be counted more than once because mutations in a gene may cause more than one phenotype and the phenotypes may be different classes. Okay. So we have done a similar exercise where for genetic eye diseases, we have found that there are a number of genes that are having two diseases, linked with two or more than, actually more than 6 diseases also. But most of them, 44% of these Mendelian eye diseases have either one or two phenotypes. But that does not restrict these whole span, which actually says about the genetic heterogeneity of these kind of rare diseases. If we look at the spectrum of human characters, this is derived from the Tom Strachan and P. Reed book of Human Molecular Genetics. You can see that there is a single gene that is Mendelian definitely. Then there is reduced penetrance, and then there are multifactorial with single major locus. So these components consists of these rare congenital diseases where you not only get the single gene with Mendelian trait, but you also get the genetic heterogeneity there also. Let's have an idea about the common and rare variants, because in the next slides we will be talking about more about this. So for these, there are 10 sequences here from 10 individuals. If you look at here, so in this position, this either A or T will be there. So that is present 3 out of 10. The A is present 3 out of 10. So that makes it a 30% probability of having this change here. So this is a common variant. On the contrary, if you look at this position, there is only one G. Rest all are C. So 1 in 10. So it's 10% only. So this is called rare variant. Okay. So if we look at the genetic architecture also, where we have this effect size plotted along y-axis and allele frequency, the rare variants that I told you about, the allele frequency, that's exactly what I have told in the previous slide. So if you look at these allele frequency versus effect size plot, you can see the rare alleles causing these Mendelian disease are here, that includes the diseases that are oligogenic. That means there are not only single gene responsible for this. There are many genetic heterogeneity that will come to the next few slides. So what is the rationale? Rationale is more than 80% of rare diseases have a genetic basis, but in majority of these cases, the causal gene is yet undiscovered and the pathogenesis is poorly understood. The vast majority of rare genetic diseases are not attributed to mutations in the known genes. Further, there has not been any uniform genotype-phenotype correlation with the rare disease-associated mutations. Intriguingly, the locus and allelic heterogeneity that I have mentioned earlier of these genes across the entire disease spectrum raises an important clinical question as to how mutations in a specific candidate attribute to multiple phenotypes. So you can see rare diseases are associated with diverse clinical and genetic heterogeneity involving tissues of multiple origin. A great majority of the rare disease associated genes regulate developmental pathways. Often mutations in the same gene result in diverse clinical phenotypes, making it very difficult for genetic counselors and clinicians to accurately counsel the patients. So I'll share some of our work regarding along this line actually. So we here in NIBMG are interested in functional genomics to delineate the genetic heterogeneity in rare diseases with neuronal defects, where we are focusing mostly on vision, hearing, and mobility. So we have models like developmental glaucoma, the diseases, congenital deafness, or congenital muscular dystrophy and myopathy. So what we do is whole exome sequencing followed by stringent filtering using bioinformatics tools and databases. And then there are two parallel things going on. One is known unknown mutations in the known candidates and prioritization of novel candidate genes using prior knowledge from database and text mining. Sanger validation, you need to do a Sanger sequencing followed by segregation analysis in the parents or siblings, and then mechanistic evaluation of mutations using appropriate molecular and biochemical assay or genetic interaction analysis in zebrafish. So that's basically the flow that we follow here for rare diseases with a lot of genetic heterogeneity, rather rare disease spectrum. So I'll share some of these, some of our results here. The first one is a story of a disease called congenital corneal anesthesia, where you can see this is a very old slide. It's published in Indian Journal of Ophthalmology in 1976. So this is a rare eye disease with hypaesthetic corneas. That means there is no sensation in the cornea. Sensory deficit either in the cornea or extended areas of trigeminal nerve endings as an isolated abnormality or part of a complex neurological syndrome. Recurrent redness and photophobia with or without associated absence of tearing. Punctuate keratopathy with persistent corneal epithelial defect. So this happens in children. So there you can see that the corneal scarring is the ultimate outcome here, and there's vision loss. So accurate initial diagnosis hence and proper management of the disease are extremely beneficial for patients to prevent vision loss due to long-term complications. If we look at the corneal structure, there are these layers where the subbasal plexus, basal epithelial, epithelium, Bowman's layer, you can see the innervations. These are the innervation and also in the stroma. Okay. So here we have done an in vivo confocal imaging in CCA patients and controls. Samples are very small, I understand that, but let me have a look what we have found. So there is a significant difference in not in their structure, but in number of these corneal and neural architecture. So if this is the control and this is the case, if you can see that the number of nerve endings are much more in control. And this is basically statistically significant, the density that we call it, not in the length, not in the thickness. So what we have done here, we have actually went ahead with a family where two individuals are affected with congenital corneal anesthesia. So we have taken the whole family, meaning this individual is our proband, that means who presented first in the clinic, and then this affected sibling, and then there is a dizygotic twin also, but which is that person is unaffected with 2 unaffected parents. So we have done whole exome sequencing. This is a huge, by doing whole exome sequencing, the number of variants can be found is 99,000 unique to all these 5 individuals. Now you need to use stringent filtering methods, and finally we came down to this gene called FAM102A. So if we look at this zebrafish embryonic eye, the structure, corneal structure, is pretty much similar with human. So we went ahead with with these expression of this FAM102A in the zebrafish, the in situ hybridization showing these FAM102A expression during development, and that is restricted in the brain and the eye region. So with that, we created mutants, the wild type. This is the wild type, and this is the mutants. The maternal zygotic mutants is created using CRISPR-Cas based genome editing. And you can see that this is basically the phenomena that we are observing in patients. We are replicating the same phenomena. You can see that the density of these nerves, this is basically stained with rabbit anti-acetylated tubulin, but you can see that the density of innervation is much less in these maternal zygotic mutants compared to the wild type. So this is basically the replication of the human phenotype in zebrafish. So another project we are basically going on in the lab currently. This is basically extension of the whatever we were doing with congenital corneal anesthesia. So this is called RED cohort, or the rare eye disease cohort, where we have a spectrum of rare diseases, includes congenital glaucoma, Axenfeld-Rieger. So these are all anterior segment diseases, anterior segment disorders in the eye. What we have done is basically again the same thing. The total number of targets were 350 genes we have screened here. 185 genes we have screened here, and 29 potential heterozygous mutations in glaucoma we have found. None of these mutations are observed in ethnically matched normal controls. The overall contribution of the candidated new genes accounted for 21.56% of heterozygous PCG cases. So this is basically the allele frequency distribution of the new genes in the RED cohort. This data is obtained from Elvita Institute, whom we are collaborating with. Next comes the another of my project, which includes the whole exome analysis of congenital muscular dystrophy and congenital myopathy patients. This reveals a wide spectrum of known and novel mutations. So these muscular dystrophies are a group of heterogeneous. So as I said, so I'm interested in the genetic heterogeneity in rare diseases, spectrum of rare diseases as such. These muscular dystrophies and myopathies are also a group of heterogeneous degenerative primary muscle disorders. This is the picture depicting how this person looks like. This is a cartoon. So what we have found is basically, I'm not going into the detail of what we have found. This is basically the number of mutations plotted based on these based on these whole exome sequencing analysis. We have done Sanger sequencing to validate our findings. These are the distributions of mutations in reported candidates. So what we have found is genetic diagnosis in CMD cases is almost 54%, and CM cases are 34% around. We have done segregation analysis. We have found some important, interesting partners in the form of desmin, where we have found this in this patient. The desmins mutations have been recognized as a cause of desmin-related myopathy. Mutations in desmin alter the coil geometry in the specific ways, leading to fatal damage to desmin filament assembly. We have found a novel stop-gain mutations identified in a congenital myopathy patients, as well as this is called lamin A. This is a nuclear filament. Mutations, we have found mutations in lamin A also. This lead to several diseases such as Emery-Dreifuss muscular dystrophy, limb girdle muscular dystrophy, and dilated cardiomyopathy. So we have found these mutations, L197P. This is a novel mutation identified in a muscular dystrophy patient. So at this point, we are looking into the mechanistic evaluation of this interaction between the desmin and the lamin, where desmin is the largest intermediate filament, and it is impaired expression can cause major alterations in myofibrillar function, whereas lamin A and lamin C are supporting components of the nuclear envelope. Specifically, these proteins are located in the nuclear lamina, a mesh-like structure of intermediate filaments and other proteins that is attached to the inner membrane of the nuclear envelope. So we are also planning to do genetic interaction analysis of lamin and despin in zebrafish during the formation of myofibrils, where we have these zebrafish embryos. You can see that these two are transgenic embryos. The first one is having MILZ2 driven. So these transgenics are having a promoter driven by a green fluorescent protein. So here the MILZ2 GFP is expressing in the muscle, and GATA1 DS-RAID is basically a vascular marker. So you can see in the raids are the raids are the vascular part, and the green are the muscle part. Similarly, this confocal image showing an F1 embryo of FLE1. FLE is again a vascular marker. And this is another transgenic fish, and KRTV8 is a epithelial cell marker. So what we are planning, we are looking at lamin and despin. We will be knocking them out using CRISPR-Cas RNP. We will do deep phenotyping, swimming pattern analysis, and formation and visualization of intermediate filaments will be observed during the developmental stages of these embryos. So I just want to share another very preliminary data. I am also interested, as I have shown earlier in my slides, that non-syndromic hearing loss or non-syndromic deafness, not going into the details of these exome analysis, what we have found is basically an interesting lead of PKHD1L1. PKHD1L1 is a stereociliary protein involved in polycystic kidney and hepatic diseases. A recent report published in Nature Communications first demonstrated its role in hearing. So what we have found is this patient having a compound heterozygous mutation in the PKHD1L1 gene. One mutation is converting the threonine to alanine in the position 2737, and the other mutation is converting a phenylalanine to leucine at 2774. So you can see these are the two mutations in one gene. This makes a perfect compound heterozygous. And then when we probed into their patients, the segregation analysis shows that the father is having mutation in one copy of the gene, and the mother is having mutation in the other copy of the gene. And unfortunately, this patient receives both the defective copy from the parents. So if you look into the conservation status of these, so this is basically where these mutations are located. We looked into the conservation status, and we can see that both of these mutations are actually conserved. The first one is totally conserved in mouse, human, and chimpanzee. Zebrafish is not very conserved. This is the serine part, but the phenylalanine mutation is the phenylalanine residue is conserved in zebrafish, mouse, and human, and chimpanzee. So I will end this with a story because my title says challenges and achievements. How the achievements can be done, I will narrate a story here. This is basically about this paper is published in Nature Medicine. It's called ARAF recurrent mutation causes central conducting lymphatic anomaly treatable with a MEK inhibitor. So what is this? The 12-year-old boy is diagnosed with a disease, a rare devastating disease spectrum of mostly unknown etiologies. So this is this lymphatic anomaly. Okay, and then what happens? So these are a few clinical parameters that you can see. The lymphangioma shows that lymphatic presence of lymphedema in several parts of his internal organs, including the inguinal areas also. These patients, they have done these. So is it not full screen? Sorry, I just thought it is full screen. Anyways, so this patient is basically having these mutations in the ARAF gene, which actually changes the residue from serine to proline, And this is also conserved in all these human, monkey, mouse, dog, elephant, and other organism. So what they have done is basically how they have found this drug. That I'm not going to in details, but what they have done is basically created transgenic zebrafish, where you have these MRC1A, where these MRC1A is basically is a marker for thoracic ductal lymphatic vessels in zebrafish with EGFP. So wherever there is lymphatic vessels, you can see this green part. And then they make they made transgenics having these ARAF. Okay, so this MRC1A derived again promoter driven ARAF, the wild type ARAF, and the mutant that the mutation they have found from whole exome sequencing. Okay, so you can see that in the thoracic duct there is a difference of distribution between the wild type and the mutant. And that is when you have superimposed with the thoracic duct, the lymphatic vessel marker, you can see that this is severe in comparison with the wild type. So now they have found a drug. This is called cobemetinib. So this drug they have introduced in these mutant zebrafish, and you can actually restore these phenotypes. Like it's exactly not normal like this one, but definitely this has the severity of these staining. You can look at these two stainings, you can see they are definitely different. And that is basically depicted in these diagram. Okay, so finally, based on this zebrafish result, they have treated this patient with this drug, and which acts as a miracle. And you can see the post-therapy lymph angiogram of this patient showing the complete reversal of this trait that was happening earlier. And these were the patient before treatment, and you can see that after treatment, the patient recovered so well. So this has been heavily cited, and this has been in the media, and the title was "The Boy Was Dying. Zebrafish helped save his life." So this, I think this is where I should end my talk. And thank you for your patient listening. And this is NIPMG. If you, when the pandemic will be over, I would like all you to visit this place. We have a fantastic genome hall inaugurated by the Nobel laureate Professor Harold Varmus. And that's it. Thank you.
Thank you, Dr. Molina, for a wonderful talk. Now, Alpana, could you please read the questions from the chat box so that Dr. Acharya can answer them?
Sure, I'm just waiting for the students or the faculty to write in, to type in a few questions there if they have any. We don't have any questions as yet.
That's good. Everybody understood everything.
Probably, yes. I think we'll just wait for a few minutes and see if they have any questions and type it in the chat box. We don't have any questions from the audience.
Okay. So I think that's it then.
Yes. So yeah. Well, if you all think of anything that you would like to ask Dr. Morinath, you can always mail it to us, and then we'll forward that information and get the answers for your questions.
Okay. Thank you all. Should I leave?
Yes, I think we can stop the meeting here then. Thank you, everyone. Thank you for joining in.